Welcome to the Krebs Cycle, the second step of cellular respiration. Also, the second step that involves at substrate level phosphorylation. Okay, so we've had two of those levels, or two of those steps. Glycolysis is the first, and then the Krebs cycle is going to generate energy by substrate level phosphorylation. So this step, again, is more about generating the electron carriers than it is about actually making energy, although we will make a little. The thing that you also have to remember here, as always, is that when we enter the Krebs cycle, we've done the link reaction, right, which is what's described here, right? Once pyruvate enters the mitochondrion, its carboxyl group is removed and is oxidized so that you're making NADPH and you attach the CoA, right? That's the link reaction that I'm describing here. So again, some of the textbooks will actually put the link reaction into the Krebs cycle. Some of them do not. But that's what we're doing here. The other important thing to remember here is we are done with glucose. We are so over glucose at this point. Once we've completed the Krebs cycle, the glucose is no more. This is the final part that's going to metabolize away any parts of the glucose that were left over. And what's going to be left at the end of this is just a bunch of molecules to restart the Krebs cycle. But the original glucose, all parts of that glucose are now gone. So this is less about the ATP generation and more about hydrogen carriers. So again, let's dig in. And I'm just showing you backbones here. I'm not trying to show you big, complicated molecules. But again, this is what we would call the link reaction here, right? This is the link reaction right there. But notice here's our acetyl-CoA coming in, and it's bonding to citrate, which is why the Krebs cycle is often called the citric acid cycle, because the first intermediate that you're bonding to creates, or actually you're bonding to oxaloacetate to create this citrate, which is citric acid. Okay, otherwise known as vitamin C. But anyway, citric acid is what you are creating here in this first step. Okay. One thing I haven't mentioned, it's been going on as we've been doing this process. It becomes really obvious here. One of the things that you're going to see happening, we've done it back in glycolysis. We didn't really make it very clear, but if you're thinking in terms of the overall equation for cellular respiration and you're trying to think about, well, wait a minute, I see the glucose coming in, and I see that water's supposed to come out, but so is carbon dioxide. So where is that happening? Well, when we strip carbon away from the sugar molecule, every time we strip it away, we end up pulling it off and releasing it as carbon dioxide. I'm not putting it in the chart because it's waste, but it is still there. Okay. And the water will get to. And then some of you are saying, wait a minute. What about the oxygen? I haven't seen oxygen. And you're right. You have not seen oxygen. And this is something I probably should have mentioned in the last video. When we get to this step right here, the pyruvate, when we get to the end of glycolysis, if there's no oxygen present, we don't generate acetyl-CoA. Okay? If there's no oxygen present, then cellular respiration stops with glycolysis. That's it. And then the cell will go through a process called fermentation, and we're going to be working through fermentation a little bit in class and explaining kind of why that happens and what its products are and so on. But notice what's really funny about that is that in order to do the Krebs cycle, in order to do this link reaction in the Krebs cycle, oxygen must be present in the cell. But as you look at this, there's nowhere where oxygen is going in yet, right? You see carbon dioxide coming off in a couple places as we strip carbons away, but there's no oxygen being used. And it's the funny part about that is it's the very last part of the very last step of cellular respiration that uses the oxygen. So again, glycolysis is anaerobic. The Krebs cycle also does not use oxygen, but it won't happen unless oxygen is present in the cell. Okay, so no oxygen is present, we stop at glycolysis. And then another metabolic process occurs called fermentation. And the outcome of that depends on what kind of organism is doing the fermenting. But then if oxygen is present, we do the link reaction and we go into the Krebs cycle, even though neither of those require oxygen in order to do them. Okay, so kind of strange thing about this. All right, so the key things to notice here are what's going in, okay, and pay attention to the direction of the arrows because sometimes it's a little difficult. We have NAD plus coming in here. We have NAD plus coming in here, and we have NAD plus coming in here, okay? So in one turn of the Krebs cycle, we can see 3 NADs coming in, and they, of course, are released as our super rock star NADHs, okay? So that's real important here, right? This is one of the big important things we're getting out of this process. I mentioned in the very first video that actually NAD plus is not the only electron carrier you're going to run into. You're going to run into a guy called FAD, also an electron carrier that creates something called FADH2. So while NAD plus can pick up 1 hydrogen, FADH2 can actually pick up 2 hydrogens. So also a super rock star in this process, okay? So let's pause for a sec, go to our chart for just a minute, okay? And I apologize, I had to handwrite these in because when I cut the chart up, you've got your chart on one nice sheet. I just cut the pieces I needed so that I could fill them out here. So I had to rewrite what each of these columns was. So again, the where is in the mitochondrial matrix, right? So we're in the matrix of the mitochondria. And so far in the Krebs cycle, what's gone in is the acetyl-CoA, right? And really, it's kind of coming out as CO2 as the waste product, okay? Because as we're stripping carbon away, that's what's left of the sugar, okay? But remember, the acetyl-CoA, there's 2 of them that are entering the Krebs cycle. So while 3 NAD pluses went in, the Krebs cycle actually turns twice for every sugar molecule because we end up with 2 acetyl-CoAs going in. That means the Krebs cycle goes around 1 time for each of them. So in reality, we don't have 3, but 6 NAD plus coming out as 6 NADH. Okay, so now we've generated 6 electron carriers. Also, again, I know you saw only 1 FAD coming in, but that's only 1 time around the Krebs cycle. The Krebs cycle runs twice, so you're going to get 2 FADH2. Okay, so we've got 8 more electron carriers. Yay, right? Oh yeah, energy. Let's go back. I'm really excited about these electron carriers, as you can tell. But we also do need a little bit of energy here. Okay, if you look down here in this bottom corner, you can see that you are going to pull off GDP is coming in. That's not gross domestic product. That's guanine diphosphate. Guanine diphosphate creates guanine triphosphate, and then that can be, the phosphate here can be pulled off to phosphorylate ADP. Why do we care about GDP and GTP? Other than the fact that that's the phosphorylated substrate that we are going to use in substrate level phosphorylation to pull off the phosphate to put it onto ADP to create ATP. So in this whole process, as you go around and you look, ATP is only created in that one little corner. And again, this goes around twice. So in reality, oops, sorry, y'all. In reality, 2 ADPs are going to come in, and we're going to end up with 2 ATP. in the process. So a little more energy. Now we've got a total of net gain of 2 ATP from glycolysis and 2 ATP from the Krebs cycle. So we've got a little more energy. But it's kind of a pittance compared to what we're going to get at the very end. Okay, so that's the Krebs cycle. That's what's important to note here from this process.